Welcome to Bioratna. I am Chandana Sengupta, and for today's session, I have selected the topic The Search for Genetic Material from the chapter Molecular Basis of Inheritance of Standard 12. So today's topic is Search for Genetic Material. Search for Genetic Material. Students, it was a common observation with the human beings that a basic set of characters always transfers from one generation to another. It is always so that the offsprings of cat are cat only, that of dog are dog only. So they are receiving some information how to form their body pattern. One basic plan is always there. And then, but if you look at the individuals of the species, then you will find that the individuals can be recognized from each other due to some variation of characters. So what is the molecule? Which molecule is responsible for carrying this information from one generation to another and at the same time causing the variation? So many scientists were working for it, and let us start first with the experiment of Griffith. Griffith, he was a British bacteriologist. He was a British bacteriologist and he was working on the bacteria Streptococcus pneumoniae. Streptococcus pneumoniae. This bacteria has two strains. One is the strain R or rough. This is the rough strain, and the another strain is S strain. And this S strain, the R strain does not have a protective covering over its body, and so whenever it enters any other organism, the host, then it cannot cause the disease pneumonia and it itself dies. But the other strain, this smooth strain, it is smooth, it looks smooth, the colonies look smooth because they have a protective covering of polysaccharide, mucus polysaccharide over their body. That protective covering cannot be destroyed by the immune system of the host. So this strain, it survives in the body of the host, causes pneumonia, due to which the host dies. This is the basic information, that this Streptococcus pneumoniae, it is responsible for causing the deadly disease pneumonia. Now, what Griffith was doing, he was actually trying to find out a vaccine for this deadly disease pneumonia. What he first did, he injected the mice with live S strain. S strain is the virulent strain. Virulent means disease causing, pathogenic strain, the smooth colonies. So it causes disease in mice and the mice dies. Next what it does, it injects the mice with live R strain. But the R strain is not virulent. It is not having the protective covering. So the R strain, it will not be able to cause the disease. So the mice survives. Next, now the only one which can cause disease is the S strain. Griffith, it kills the S strain. So the mice is not now injected with it killed S strain. The S strain which can cause disease, it has died. When it has died, it cannot cause the disease. So now the mice survives. Fine. But the fourth experiment, this one was having some different result. What was it? Now the mice was injected with a combination of two which cannot cause the disease. That means the dead S and the live R. लाइव आर जो है वो जिंदा है लेकिन वो डिजीज कॉज नहीं कर सकता है. एस जो है वो डिजीज कॉज कर सकता है बट इट इटसेल्फ इज डेड. सो द एक्सपेक्टेशन इज दैट द माइस हैज टू सर्वाइव. But to his surprise, what he finds out is the mice now has pneumonia and it dies. How this happens? The S strain which can cause disease has already died. It cannot cause disease, that we have verified here in this third experiment. Heat kill S cannot cause disease. The live R also cannot cause disease. So whatever is being done, that has to be done by the living one. जो जिंदा है, कौन जिंदा है? The rough strain, R strain, which is non-virulent one, has suddenly become virulent. So now comes Griffith conclusion. He couldn't find out the molecule due to which the R strain transformed itself into S1, how it got changed. But his natural conclusion, called Griffith's transforming principle, it states that something from the heat killed S strain, it passed into the R strain and made the R strain virulent. जिसकी वजह से वो R strain virulent हो गया because now it got the information how to form that protective covering. That molecule which carried this information, which was having this information, which the information S1 was having, R1 was not having. Now the R1 has received that information and has become virulent. But Griffith could not find out that molecule, which molecule is responsible for this. But his experiment paved way for the other scientists, that now they started working what was that molecule which had that information and which could change the R strain into S strain. So we will come to that next experiment by Avery, MacCarty and McLeod. Now the experiment by Avery, McCarty and MacLeod. They repeated the experiment of Griffith and they tried to find out the biochemical nature of Griffith's transforming principle. वो मॉलिक्यूल जिसने R strain को S बना दिया था, वो इंफॉर्मेशन जिस मॉलिक्यूल में था, जिस मॉलिक्यूल को ग्रिफिथ खोज नहीं पाया था, now these three they worked upon it to find out its biochemical nature. What they did, from the A strain, from the virulent strain, they purified RNA, protein and DNA. Now, one by one, they introduced RNA, protein and DNA into the R strain to see which one converts R into S. When RNA and protein were introduced, then there was no transformation. But when DNA was introduced, then the R strain changed into S strain. Fine. This is one experiment. They did another experiment. What they did, now this RNA, it was destroyed with the help of the enzyme RNase, but they found out that destroying RNA does not have any impact on transformation. The R still can change into S. Similarly, when this protein, it was destroyed with the help of the enzyme protease, still it had no effect on transformation and the R could still change into S. But when this DNA was destroyed with the help of enzyme DNAs, जब DNA को destroy किया गया, then there was no transformation. The R strain could not be changed into S. So these two experiments were by them to prove that DNA is the genetic material. Do you think that they successfully proved that DNA is genetic material? No. Let me tell you that at that time, it was protein which was thought to be the genetic material. Why so? DNA has only 4 variations in its nitrogen bases, A, T, C and G. So the natural logic was that how can a molecule having only 4 variations can control so many characters, whereas there were so many proteins which could successfully control the different types of characters. So it was thought that it is the protein contamination in the DNA. Protein contamination in the DNA which was responsible for the transformation. Even after this wonderful experiment by Avery, McLeod and MacCarty, DNA was still not recognized as the genetic material. There was a logic behind it. DNA is made up of 3 types of molecules. One is a pentose sugar, one is a phosphate group, and there is nitrogen base. There is no variation in sugar and phosphate group. All sugars of DNA are same, all phosphate groups are same. There is variation in only nitrogen base. That too, there are only 4 types of nitrogen bases. So it was a logical thinking that how can a molecule which varies in only 4 nitrogen bases can control so many different types of characters, whereas protein, so many different types of protein were naturally thought to be the genetic material. Little did they know that yes, these are the proteins which carry out the work, but the information, even how the proteins are to be formed, that information, what work they are going to do, this whole information is there in the molecule, that is the DNA. But now here it is not proved what they thought. Then what conclusion they gave, what the other scientists at that time they thought that it is the contamination in DNA, not the DNA has carried out the transformation, but the DNA must be contaminated with protein, and that protein has carried out the transformation. So next step has to be taken. Someone else has to conclusively prove that it is DNA and not contamination of protein which brings out that transformation. Now, let us see the experiment by Hershey and Chase, which will conclusively prove without any doubt that it is really DNA which is the genetic material. So, Hershey and Chase, who published their reports in 1952, their experiment was on E. coli and bacteriophage. This bacteriophage is a type of virus which infects the bacteria. It has a simple structure. It has the body made up of a protein coat. This whole body, it is made up of, this one is the protein, and inside it has the genetic material which is the DNA. So it is having both the contenders, proteins as well as DNA. So Hershey and Chase selected this bacteriophage. Now what they did, they prepared 2 sets of bacteriophage. One they grew in P32, another is S35. P32 and S35 both are radioactive. P32 and S35 both are radioactive. One thing is that, as you know, P is present in DNA. It is present in DNA, but it is not present in protein. P32 is present in DNA, not P. Phosphorus is present in DNA, not in protein. And sulfur is present in protein. It is present in protein, yes, but it is not present in DNA. Okay. The normal P and S are present in DNA and protein. Here they are using the radioactive one so that we can test it and find out which one is the genetic material. Now, when P32 is used, so naturally the DNA will be incorporated with radioactivity and the protein will not, because protein में phosphorus है ही नहीं, तो P32 वो लेगा ही नहीं. So what will happen if this is the bacteriophage and this is its DNA? So the DNA will become radioactive. This one will become radioactive, but the protein will remain non-radioactive. This is the one bacteriophage. Another set which they prepared, this bacteriophage, which is grown in S35. So S is present in protein in two amino acids actually, cysteine and methionine. One is cysteine and the another is methionine. These two amino acids, they have the sulfur. Now, here the protein will become radioactive because proteins have sulfur, but the DNA will remain non-radioactive as DNA is not having any sulfur. Now this is made to infect the bacteria. When it is made to infect the bacteria, here they infect, here also they infect. Then what will happen? It infects the bacteria. Here also it is infecting the bacteria. When it infects, what it does? It introduces its DNA into the bacteria. It introduces its DNA into the bacteria. Here the DNA will be non-radioactive, protein will be radioactive, and here the DNA will be radioactive, but the protein will be not. Now, this is blended. Why? This is blended to separate. This bacteriophage is attached to the body of bacteria with the help of these tail fibers. With the help of these tail fibers like a claw. So now we blend it so that it gets agitated and it gets separated from the body. So now it will be separated and then it is centrifuged. After that there is centrifugation also, and when centrifugation, then this two will form different layers, and here the protein will be radioactive, here the DNA is radioactive. Let me draw the DNA of bacteria also here. Okay. Now you see the simplicity of the experiment. What was their whole planning was based on the fact that the proteins are not having P and the DNA is not having S. Now when they are grown in bacteriophage in S, then they become radioactive, the protein becomes radioactive, but as the protein does not enter into the bacteria, so it remains outside, so protein is not the genetic material. When P32 is used, P is present in DNA, so the DNA becomes radioactive, it enters the bacteria, and radioactivity is found in bacteria. So now conclusively it is proven that the protein is not the genetic material, but DNA is the genetic material. So the journey we started in 1928 by Griffith, successfully completed in 1952 by Hershey and Chase. Here ends the search for who the genetic material is. Now we'll start the search for the structure of DNA. Join now, subscribe. See you in the next meeting.